Welcome back everyone to the final part of this pharmacodynamics topic, and it is on drug targets. So you can see that there's only 3 learning objectives for this part. Now, as I mentioned much earlier on in the topic, there are 4 main targets for drug action. We have receptors, we have ion channels, we also have enzymes, and of course we also have those transport or carrier proteins. We just go back here to the receptors. What are the receptors? These are the sensing elements in the system of chemical communications, and they coordinate the function of all different cells in the body, including hormones binding to receptors, neurotransmitters binding as well. And the example we've given you here is the histamine binding to the histamine receptor will produce a particular effect. And also we have the beta adrenergic receptors, and there's heaps more, receptors. Many of them are on the cell surface, some of them, so extracellular receptors, some of them are inside the cell, so intracellular receptors, and they're often nuclear receptors. Now, looking over at this little schematic here, you can see that we've got our receptor. Our little pink ball there is our agonist, or it might be an inverse agonist. Now, we don't talk too much about inverse agonists, but literally an inverse agonist is a drug that binds to the same receptor as the agonist but produces the opposite effect. Okay, easier to give you an example. Let's say in the case of benzodiazepines, they bind to receptors, and the net effect of them binding will be to cause some sedation, right? You could use then an inverse agonist then, which would kind of have the opposite effect and cause things like anxiety or convulsions, but we don't talk about them in this subject, so let's leave it at that. Okay, so the agonist binds to the receptor, and there's either a direct effect or signal transduction mechanisms that occur. But some of the net effects then of the agonist binding to the receptor are opening and closing of ion channels, or enzyme activation, or even inhibition of an enzyme, ion channel modulation as well, and DNA transcription too. And that obviously will be an intracellular receptor that causes that DNA transcription, which is going to eventually lead to protein synthesis. You can see we've got our antagonist here, or our inhibitor, is with this dark grey ball, and when it binds to the receptor, remember, it displays affinity but no efficacy, so there will be no effect, but by its presence being there, the endogenous mediators or ligands are blocked. The second target are these ion channels. And what are ion channels? They are essential gateways in cell or plasma membranes which selectively allow passage of particular ions. For example, local anesthetics block voltage gated sodium channels. If you look over here, you can see the ion channel with the blocker in situ, so the drug molecule in situ, and while it's there, this channel is blocked, so the flow or the permeation of ions is inhibited. We've also got the action of a diuretic called amiloride, and what it does is it acts in the kidneys, in the nephron tubules, if that word is familiar to you, to block sodium entry into the renal tubular cells, right? So as a result of this drug, sodium stays in the renal tubules, and that's going to increase the osmotic pressure of the filtrate and draw more water into the filtrate, and the person is going to then excrete higher volumes of urine, which will lower the blood volume and lower the blood pressure. So this drug is often given for somebody with hypertension. You can also have these modulators, and a modulator in terms of a drug here is altering or adjusting the activity of this ion channel. Right, so there'll be either increased or decreased opening probability of an ion channel through the use of particular modulators. The third target then are enzymes, and of course enzymes are biological catalysts that regulate the speed of chemical reactions and therefore regulate our metabolism. And often a drug molecule is a substrate analog that acts as a competitive inhibitor of the enzyme. So that's what you're looking at here in terms of this little drug molecule here. It's acting as an inhibitor, and the normal reaction is then inhibited. We've given you 3 examples here, but you don't need to remember the names of them at this stage. We've got captopril. That one will come up in the cardiovascular topic as it's given to reduce blood pressure. Neostigmine, that will come up in the immune system topic. And aspirin, acting on this enzyme here, cyclooxygenase, will come up in the anti-inflammatory topic. Now, other things that can happen is you could use, a false substrate, right? And that false substrate is competing with the normal substrate for the binding site. And in doing that, an abnormal metabolite is then produced. So that will be that kind of pharmacodynamics of that particular drug. Or, enzymes are important in activating prodrugs into active drugs. And then finally, looking at these transport proteins or carrier proteins that span the plasma membrane. You're looking at one here, and they allow ions and small organic molecules to pass through lipid molecules. So here you can see an agonist or substrate binding there, and it's then, transported from the extracellular side to the intracellularly. Okay, so you can inhibit these transport proteins, and here you've got an example of that. This drug is inhibiting this transporter here, and therefore transport of the agonist or substrate is therefore blocked. Or you could use a false substrate, okay? And in that case, the false substrate then is the one that is, sort of accumulated on the intracellular side. And an example here of a class of drug that inhibits these transporters are the tricyclic antidepressants, which act on the noradrenaline transporter. So they block the noradrenaline transporter, and in doing that, they block that noradrenaline reuptake mechanism. Now, just like we have 4 main targets for drug action, there are also 4 main types of receptor proteins. The ligand-gated ion channels, the G protein coupled receptors, and the kinase linked receptors are cell surface receptors. Okay, so they will interact with water-soluble ligands or drug molecules that can't cross the lipid bilayer. And then the 4th one are the nuclear receptors, and they're obviously found inside cells. So these are intracellular receptors. Okay, this slide's in a lot of detail, everybody. We don't expect you to memorize it all at this stage. But when you are researching for assessment 2, which is your presentation, you may be talking about a couple of these different receptor types, in terms of the drugs that you choose. Okay, so starting with these ligand-gated ion channels, they're also known as ionotropic receptors here. So we've got our receptor, we've got our drug molecule, our agonist binding there, and that will then cause action on an ion channel to either open it or close it, leading to things like hyperpolarization or depolarization, depending on the direction of flow of ions. But it is that flow of ions and that produces that cellular effect, okay? The response time to this binding is super quick, right? So it's on a time scale of milliseconds. You don't need to remember this example here, but the nicotinic acetylcholine receptor is an example. The second one are these G protein coupled receptors, also known as metabotropic receptors. There's a couple of different things that can happen here. Again, we've got this cell surface receptor with our agonist binding, activating or reducing the effect of a G protein to sort of have an effect there on an ion channel. Again, that will cause a change in excitability, which will lead to a cellular effect. The second thing that can happen, though, is this sort of, I guess, signal transduction sort of mechanism that can occur. So again, the agonist binds to the receptor, activation of this G protein, which then leads on to that sort of maybe enzymatic sort of process, that second messenger process might lead to protein phosphorylation or other things that lead to the cellular effect. And that cellular effect is in response to that agonist binding to that G protein coupled receptor. The third one is the kinase linked receptor. Wherever you see kinase, you know it's an enzyme, and it's an enzyme that adds phosphates to something. So again, we have our agonist binding causing protein phosphorylation, leading to gene transcription, and we know that once a gene is transcribed, that's then going to lead to messenger RNA synthesis, and then that messenger RNA is going to be translated into protein synthesis, and that's the cellular effect there. And for these nuclear receptors, because they are intracellular, it's only really lipid soluble agonists that can freely dissolve and flow through the phospholipid bilayer. So the steroid hormones, for example. An example here is the estrogen receptor. So binding there of the agonist to that estrogen receptor, leading to gene transcription, protein synthesis then, which is the cellular effect. Now, if we just go, I said the response to for the ligand-gated ion channels is super quick. It's also very quick in terms of the G protein coupled receptor response, slower than the first one, but still seconds. Then we have the other two, which are much slower. So hours rather than seconds. Example of the kinase linked one is a cytokine receptor, and as I mentioned, the nuclear receptor there being the estrogen receptor. Okay, when you're almost at the end, just looking at control of receptor expression now. Firstly, we have desensitization. Remember, we talked all about that earlier on, and all of the sort of mechanisms of desensitization for short-term regulation of receptor function. So things like those conformational changes in the receptor, etc. Also up or down regulation. So remember, we talked about that, how upregulation of receptors is that there's more receptors for an agonist to bind to, or the endogenous ligand, and that's for long-term regulation, as is down regulation as well, where the number of receptors available are reduced. So there's fewer receptors for the agonist or ligand to bind to. And an example of this makes it a lot easier. So some viruses will cause upregulation of growth factor receptors. So the more growth factor receptors that are available, the more growth factor that can bind to those receptors. And unfortunately, when this happens, this can lead to cancer, cells becoming sort of immortalized due to that, constant stimulus of the growth factor and the availability of those growth factor receptors. It can lead to cancer in genetically predisposed people. And so a number of diseases are directly related to receptor malfunction. Very briefly here, this autoimmune condition called myasthenia gravis, which is a progressive autoimmune condition that leads to progressive muscle wastage. We will talk about this in the immune topic, so don't worry, you don't need to know that much about it at the minute. But what happens with this disease is the immune system produces an autoantibody, and that autoantibody is directed directly against the receptor. Right? So this autoantibody will inactivate the acetylcholine receptor, and this is what leads to that progressive muscle wastage. The binding of the autoantibody to the receptor will either destroy the receptor or inactivate it or cause it to be internalized. Regardless, there's less receptors, or there's fewer receptors for the acetylcholine to bind to, and therefore the muscle wastage that follows. And mutations in genes encoding receptors and proteins involved in signal transduction. For example, mutation of thyrotropin. Now, if you remember back to your physiology, thyrotropin is another word for thyroid stimulating hormone. And thyroid stimulating hormone binds to cells in the thyroid gland that synthesize and secrete T3 and T4, which are your thyroid hormones. So if you have a situation where there's a mutation of the thyrotropin gene, then you're going to get continuous oversecretion of thyroid hormone, and that then leads to the symptoms of hyperthyroidism. Okay, everybody, we have finished. Thanks for bearing with me, and when I come back, we're going to be moving on to pharmacokinetics.